In this video, we will discuss about different types of Siglec and Sialoglycans binding or their interactions. So, this Siglec and Sialoglycan interactions can occur in the form that these Sialoglycans or the ligands for the Siglec can be in the form of like a soluble form, which has been secreted by the tumor cells, or they can be expressed on the tumor cell surface that is shown here, like this glycoprotein, or they can be secreted in the form of extracellular vesicles that interact with the Siglec that are expressed on the immune cells. So these interactions can be in the form of three different major forms, either in the soluble form or on the cell surface, or in the form of extracellular vesicles. Now let's start with these different types of Siglec and Sialoglycan interactions. First one is this trans-binding. The trans-binding means that the Siglec and its ligand or Sialoglycans are from two different sources. So in this case, the Sialoglycans can be is from the tumors. When once it interacts with this Siglec, whether it's inhibitory Siglec or activating Siglec, it will trigger the corresponding signals. And in this case, the activating signals are usually a positive charge, whereas inhibitory signals does not have that positive charge. The next type of interactions is this cis-binding. So in the cis-binding, what happens? The source of the Sialoglycans and Siglecs are from the same cell surface. So here it is, as it is shown in this figure, that the Siglecs are on the immune cells and they interact with the sialic acid based Sialoglycans on the same surface. So in this case, it can either interact, depending on the specificity, it can interact with this inhibitory Siglec or the activating signal. However, sometimes it has been reported that there is a dynamic change between cis-binding and trans-binding. However, the mechanism is not yet been known how this dynamic change occurs and what derives this dynamic change from cis-binding to trans-binding or trans-binding to cis-binding. The third type of interactions is called as the clustering interactions. In this case, when in the tumor cells, as it is very common that there is a cluster of Sialoglycans formed, because in the tumor cells, as we have seen, that there is in most of the cases there is a oversilation occurs, and this cluster Sialoglycans once interact with this immune cells, and we have seen that in the immune cells, especially when they are in tumor microenvironment, there is overexpression of the Siglec. So this makes a favorable conditions for this clustering, where clustered Sialoglycans interact with this clustered Siglec, and it will trigger a immune response. In this case, since it is a inhibitory Siglec, it will trigger enhanced immune suppression, and thereby this kind of interactions helps the tumor cells in immune evasions. And as we have seen in one of the paper, where the ligands on the pancreatic cells interacts with the Siglec-10 receptors on the macrophages, this will not only this will not only prevent the phagocytosis of the this myeloid derived suppressor cells towards this tumor, but at the same time it will also prevent the activations of the other immune cells, such as activations of the T cells by providing antigen information. So those things will also get suppressed. So this kind of interactions might have a cascade effects depending upon what kind of interactions it occurs. If it is inhibitory signals, since from one point it will cause enhanced immune suppressions because of the multiple interactions. At the same time, it will also reduce the activations of the T cells by reducing the antigen presenting informations to the T cells. So these are three major ways or three major classifications of the Siglec and Sialoglycans, and how they might affect the immune response. Whether it might activate, if it is activating Siglec, it might inhibit if it is inhibitory Siglec. So I hope you understood to some extent this Siglec and Sialoglycans binding. If you need to understand more about this topic in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.